Hey everyone, and welcome back for another deep dive. Today we're going to be looking at a pretty amazing discovery in canine neurology.
Oh, yeah.
It's researchers have found a new genetic variant in miniature American Shepherds, and it's linked to neuroaxonal dystrophy, or NAD.
You got it.
And this is really cool because it actually closely resembles a human condition called SPG85.
Right.
So we're talking about a potential for some really big breakthroughs.
Yeah, absolutely.
In terms of treatment and therapies for both species. And our main source for this deep dive is a research article from the Movement Disorders Journal.
Okay.
It's called Canine RNF170, Single-Base Deletion in a Naturally Occurring Model for Human Neuroaxonal Dystrophy.
Wow, that's a mouthful.
Yeah, it is. But
very descriptive.
yeah, it's packed with a lot of really fascinating details.
Yeah, and I think what's really interesting about this is what you were saying about the connection between canine and human neurology.
Right.
That's really, I think, where the big excitement is because we can learn so much from each other.
Absolutely. So for those who aren't familiar with NAD, this condition causes swellings called spheroids to form in the axons of the central nervous system.
Okay.
So you can kind of think of it like kinks in a hose.
Yeah.
It disrupts the flow of information along the nerves.
Right.
And while NAD is found in several dog breeds, this finding in miniature American Shepherds is particularly groundbreaking
I see.
because it mirrors the human SPG85 condition so closely.
Okay.
So we basically have a naturally occurring model to study this disease in humans,
Right.
which is huge for research.
Yeah, absolutely. And the research team really took a comprehensive approach to figure all of this out.
Oh, yeah.
They combined, clinical examinations, so looking at the dog's behavior and symptoms,
Right.
with pathological investigations, which included necropsies to examine the affected brain tissue.
Yeah, and to pinpoint the genetic culprit, they went all out.
Oh, yeah?
They did GWAS autozygosity mapping and even whole genome sequencing.
Wow.
It's like they brought out the big guns of genetic analysis.
Yeah.
Yeah.
They really did.
Yeah, and so they found that one of the most common symptoms they observed in affected miniature American Shepherds
Yeah.
was hind limb weakness and ataxia, which led to this kind of unusual gait
Interesting.
where the dogs move both legs on the same side of the body together,
Oh, wow.
almost like a camel walk. Have you ever seen videos of that?
Yes, I have. It's very distinctive.
It is.
You can't miss it.
Right.
And then on top of that, they also noticed some other things.
Okay.
Like scuffing of the paws,
Uh-huh.
kyphosis, which is kind of an abnormal curvature of the spine,
Gotcha.
and even a gait that suggested that the cerebellum was involved.
So like a range of neurological issues.
Yeah, it really does seem that way.
And did the necropsy findings match up with these symptoms?
Well, that's a great question, and
Did they find those spheroids in the brain tissue?
they did.
Okay.
Yeah, the necropsies of 2 affected dogs revealed a significant number of spheroids, particularly in the brainstem. And they also found evidence of neuronal degeneration and gliosis, which is an increase in glial cells.
And those are the support cells of the nervous system.
Exactly.
So all signs point to NAD.
It seems that way.
But what about the genetic culprit?
Right.
Did they pinpoint the exact genetic change causing all of this?
They did. They found a single base pair deletion in a gene called RNF170.
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 Okay.
And this deletion leads to what's known as a frame shift mutation.
Okay, a frame shift.
Yes.
What is that?
Well, so think of a gene's DNA sequence like a blueprint
Yeah.
with 3-letter words, each representing an amino acid, the building blocks of proteins.
Okay.
Now imagine deleting a single letter from one of those words.
Right.
It throws off the entire reading frame like scrambling a sentence.
Oh, sure.
That's a frame shift mutation.
So instead of building the correct sequence of amino acids,
Right.
the cell ends up with kind of a jumbled mess,
Exactly.
leading to a non-functional protein.
Yeah, it's like trying to build a house from a blueprint. With missing or scrambled instructions, you're not going to get the outcome that you want.
That's a great analogy.
Yeah.
So this tiny deletion in the RNF170 gene essentially sabotages the production of the RNF170 protein.
Right.
What's the big deal about this protein?
Well, this is really interesting.
Okay.
RNF170 is an E3 ubiquitin ligase,
Okay.
and it plays a crucial role in regulating these receptors called IP3Rs,
Okay.
which control the flow of calcium within cells.
Wait, calcium, like what we need for strong bone?
Yes, the very same calcium.
What's it doing in our nervous system?
Well, calcium is a really versatile signaling molecule.
Okay.
And it's involved in tons of cellular processes, including nerve impulse transmission.
I see.
And this is where it gets really interesting because Variants in the RNF170 gene in humans have been linked to conditions like HSP and SNAX1, both involving motor impairments similar to what we're seeing in these dogs.
Wow, this is way bigger than just dogs.
Absolutely.
These miniature American Shepherds could actually be like a living, breathing model.
Exactly.
For studying RNF170-related conditions in humans.
You got it.
That's incredible.
It really is. And because these dogs have a relatively long lifespan, it opens up so many opportunities for long-term observation and therapeutic trials.
Oh, yeah.
This is really a game changer.
It's huge.
For research in both canine and human neurology.
We're talking about potentially unlocking new treatments for these debilitating neurological conditions, all because of this one little genetic glitch.
Yeah, one little genetic glitch that opened up a whole world of possibilities.
This is amazing.
Yeah.
All right, so we've got to talk about the implications of this discovery, particularly for miniature American Shepherd breeders.
Right, because now, if this genetic variant is out there, how do we prevent more dogs from being affected?
That's a good question.
It is, and I think genetic testing is going to play a huge role here.
Yeah, for sure.
Now that we know about this specific deletion, breeders can test their dogs to see if they carry this mutation and make informed decisions about breeding.
This is going to be key information.
It's absolutely.
For breeders and owners.
It really is.
Wow, we've covered a lot of ground here, but I'm ready to dig even deeper into the implications of all of this.
Me too. I think there's so much more to unpack here, and I think our listeners are going to find it fascinating.
I think so too.
One of the things that really struck me about this research is how it showcases the power of comparative medicine.
Yeah.
You know, by studying diseases in animals.
Right.
We can gain such valuable insights into human health.
Absolutely. It's like we have this natural experiment happening right in front of us.
Yeah, exactly.
And it's giving us clues about this group of really devastating human neurological disorders.
Right. And in this study, the researchers actually used a very specific technique to find the genetic cause.
Okay.
It was called autozygosity mapping.
Oh, yeah. Autozygosity mapping. Can you remind us what that's all about?
Sure. So it's a technique that we use to identify genes that are associated with recessive diseases. And it works because in small, closed populations, like some dog breeds, there's a higher chance of inheriting identical gene copies from both parents.
So by tracing these identical gene segments in affected dogs,
right,
researchers can zero in on the location of the disease-causing gene.
Exactly. It's like having a genetic magnifying glass.
Oh, I like that.
Leading them straight to the RNF170 gene.
Okay, that's pretty cool. But I gotta admit, I'm still a little fuzzy on how that single base pair deletion actually impacts the protein.
Sure.
Can we go back to that frame shift idea for a sec?
Of course. So remember, a gene's DNA sequence is like a blueprint for assembling a protein. And each three-letter word in that blueprint, called a codon, specifies a particular amino acid.
Right. So the codons are like the instructions for building the protein.
Yes. And now imagine if you delete a single letter from a sentence.
Okay.
The rest of the sentence becomes gibberish.
Yeah.
Same thing happens with a frame shift mutation. That single base pair deletion in the RNF170 gene shifts the reading frame, and it alters all the codons that come after it.
So instead of the correct sequence of amino acids, the cell ends up with a jumbled mess.
Exactly.
Then leads to a non-functional protein.
Right. It's like trying to build a house from a blueprint with scrambled instructions.
Yeah. You're not going to have a good time.
Exactly. A structural disaster.
Yeah. So in this case, that structural disaster is a non-functional RNF170 protein.
Yes.
That can't regulate those IP3 receptors, leading to all the neurological problems we see in NAD.
It's a cascade of events, all stemming from that one tiny deletion in the DNA.
It's amazing how such a small change can have such a huge impact. Did the researchers have any idea when this mutation might have first appeared?
They did.
Okay.
They actually estimated the age of the mutation to be around 27 years old.
Wow, that's pretty recent.
Yeah, in evolutionary terms, that's very recent.
So this mutation probably emerged after the miniature American Shepherd breed was established.
Exactly. It's not some ancient mutation that's been lurking in the breed's lineage for centuries.
Wow, so that raises some questions about how this mutation became so common.
It does.
You know, without genetic testing, breeders might have been unknowingly selecting for this mutation.
It's definitely possible, and it highlights why genetic testing is so important for making informed breeding decisions.
To prevent these harmful mutations from spreading.
Exactly.
So speaking of which, do we know how common this mutant allele is in the miniature American Shepherd population?
Well, the researchers looked at a large group of miniature American Shepherds and found that the mutant allele frequency was 8.3%.
Okay.
And that translates to a carrier frequency of 16.6%.
So roughly 1 in 6 miniature American Shepherds carry this mutation.
That's right.
Wow, that really shows how important genetic testing is.
Absolutely. Knowing a dog's genetic status is crucial for making those informed breeding choices.
Right, and reducing the risk of producing affected puppies.
It's about empowering breeders to protect the health of their lines.
Exactly. And for potential owners, it means asking breeders about genetic testing.
Yes. Responsible breeders should be transparent about their testing practices.
We've talked a lot about preventing NAD, but what about the dogs who are already affected?
Right.
Is there any hope for treatment?
That's the crux of the matter.
Yeah.
Unfortunately, there's no easy answer. There's no cure for NAD in dogs or humans.
Right.
But this research is really valuable because it gives us a naturally occurring model to study how the disease progresses and to test potential treatments.
Thus, we can monitor how the disease progresses in these dogs and see how different therapies affect them.
Right.
And hopefully translate those findings into human clinical trials.
Exactly. These miniature American Shepherds are like pioneers, paving the way for treatments that could help both dogs and humans.
It's really amazing how much we can learn from our four-legged friends.
It is.
Their contribution to science is remarkable. Yeah. But the researchers mentioned that the clinical signs in these dogs, especially the later onset and the potential involvement of other neurological systems, might resemble another human condition called SNAX1.
That's a great observation.
Yeah.
SNAX1 stands for sensory ataxia neuropathy with vestibular arflesia.
Okay.
It's a rare neurological disorder that affects balance coordination and sensory perception.
So these miniature American Shepherds could be a model for two different human conditions.
It's incredible, isn't it?
Yeah, it makes this research even more valuable.
It's like a 2-for-1 deal.
Yeah, 2 for 1.
In terms of scientific insight.
It sounds like there's still so much to learn about this gene and its role in neurological health.
Absolutely. This is just the tip of the iceberg.
Wow.
But, you know, we've talked about the importance of genetic testing for breeding decisions.
Right.
But there's another important aspect to consider, and that's genetic counseling.
Yeah. What does genetic counseling involve?
It's a process of providing information and support to people and families who are either at risk for or affected by genetic disorders.
So in the context of miniature American Shepherds, a genetic counselor could help breeders understand the inheritance patterns of this mutation, the risks, and their breeding options.
Exactly. And for owners of affected dogs, a genetic counselor can provide valuable information about the disease, its progression, and how to manage it.
So it's about connecting the complexities of genetics with the real-life decisions that breeders and owners face.
You got it. Genetic testing is just one piece of the puzzle.
Okay.
Genetic counseling puts all the pieces together and helps make sense of it all.
It sounds like genetic counseling can really help people make the best choices for their dogs and for the breed as a whole.
Absolutely. Knowledge is power.
This is incredible to think about how this one research article could impact not just canine neurology, but also human health.
Absolutely. And there's one more thing I wanted to go back to in the research, because I think it's really important for understanding how NAD works. Remember we talked about the ubiquitin proteasome system? Well, I think it's worth taking a closer look at that.
Yeah, it is a pretty complex system. I'd love to dive a little deeper into that.
So just imagine it as like a cellular recycling plant. It's responsible for breaking down and removing all those damaged proteins. And the unwanted proteins keeping everything running smoothly.
So it's like the cell's own little garbage disposal.
Yeah, exactly. And ubiquitin is a small protein that acts like a tag. It attaches to proteins that need to be recycled.
So it's like putting a sticky note on a file that says, shred this.
Exactly.
So ubiquitin marks the protein for destruction. What happens next?
Well, that's where the proteasome comes in. This is a complex molecular machine that recognizes those ubiquitin-tagged proteins and breaks them down into smaller pieces.
So the proteasome is the shredder.
Exactly.
Making sure that those unwanted proteins are gone for good.
Gone.
And where does RNF170 fit into all of this?
Well, RNF170 is an enzyme called an E3 ubiquitin ligase. And these enzymes are like matchmakers. They bring ubiquitin and the protein together.
So RNF170 helps shred this note onto the right protein.
Exactly.
Which in this case is that IP3 receptor.
Yes, but when the RNF170 gene has that deletion, it can't tag the IP3 receptor for destruction.
Okay.
And then they build up,
Uh-huh.
and it messes up the calcium signaling.
Right, leading to those neurological problems.
Yeah, it's pretty incredible how this one little genetic flaw can have such a big impact.
It really is. And what I find so exciting about all of this is that it's not just about understanding the disease.
Right.
It's about finding new treatments that could improve lives.
Absolutely.
This research is giving us a really solid foundation to build on.
It is.
Well, I have to say I've learned so much from this deep dive.
It's been a pleasure.
It's been fascinating to
Okay.
really get into canine genetics and see how it connects to human health.
Yeah.
Before we wrap up, do you have any final thoughts for our listeners?
Well, I think the big takeaway here is that the more we know about genetics, the better decisions we can make
Yeah,
about our dog's health.
that's a really important message, and it makes me wonder what other discoveries are out there waiting for us.
I know, it's really exciting.
It is. All right, well, a huge thank you to our expert for joining us today for this incredible deep dive.
Thank you for having me.
And to all our listeners out there, thank you for being here with us. From Vet Neurojar, keep those minds inspired, hearts light and tails wagging.